This is our interview for the cell structure and function.
So then this green thing right here is a chloroplast. This is found in plants.
Yeah.
This is found in plants, and it takes in sunlight,
Carbon dioxide,
Carbon dioxide, which
water,
is here.
and it
Water. These. Okay, so these takes in these two plus sunlight.
put sunlight energy,
and it turns
and it
to
yields
energy.
to
ATP.
no, not ATP.
A little bit of ATP.
A little bit of ATP.
Glucose and oxygen.
Sugar.
Yeah, and then. Yeah. All right.
And
It's
then,
called photosynthesis.
Yeah.
Mm-hmm.
Because
And then we have this red thing, which is the mitochondria,
yeah.
the powerhouse of the cell.
No, it is not.
No.
We've been lied to. It is not the powerhouse of the cell.
Wow.
It-
Okay, so the mitochondria does this process called cellular respiration,
Yeah.
and it takes in oxygen, glucose, and energy.
No, it does
Or
not.
not
It
energy.
yields energy.
Yields energy.
It yields ATP. It
And-
turns.
and
It turns
carbon because
And
we-
carbon dioxide.
and carbon dioxide. Yeah. So it turns ADP into ATP. It adds a phosphate to the
Yeah.
end.
So that's.
And then we have lysosomes, peroxisomes, and regular old vacuoles. So lysosomes are basically just membrane sacs filled with enzymes that digest like cell food, and so it just digests
And
for
they
energy.
also digest broken organelles.
Yeah. And then peroxisomes digest. Peroxisomes have a crystalline core and they digest fats.
and then vacuoles.
And then
are-
hold on. Peroxisomes
Fine.
decide if fats are going to be used for other, for membrane or if it's used for energy.
Mhm.
And they also take H2O to oxygen, and then it makes hydrogen peroxide, which then turns into H2O, which is water.
H2O2.
And now to vacuoles, and it's just storage.
Yeah.
Yeah.
It's just storage.
It's membrane sac.
Yeah. It's just there. Okay. Next, protein synthesis. So protein synthesis is basically just the process of
Making proteins.
because synthesis means to make.
Yes.
Okay.
So this is the nucleus. The nucleus has the nuclear envelope, which is a double.
Double phospholipid bilayer.
Yeah.
And then the nucleus controls the cell and stores the DNA.
So
This is the DNA.
Yeah.
This is DNA because it's double stranded.
Yes.
Yeah.
This is
And then
RNA.
RNA is single-stranded DNA.
So what happens is the DNA, the helix unzips, so then the RNA comes along and is able to copy, to copy the code.
and so this is
mRNA.
mRNA, messenger RNA. It comes out of the nuclear envelope and it enters a ribosome. Not enters, but like it comes into contact with a ribosome.
For the purpose of this demonstration, we're going to make it go through the endoplasmic reticulum,
Okay,
The
so,
ER.
the ER.
So
The ribosome's attached to the ER, so it's going to go through the ribosome into the ER.
and so
And it reads it,
the ribosome is going to read it one triple at a time, which is one codon.
and then
Yeah.
it adds in.
Amino acids.
Yeah,
Yeah.
depending on what it reads.
Yeah.
Yeah.
And then that protein chain goes through the endoplasmic reticulum, and then once it gets to an end, it could
It's
gonna bunch off, and then it becomes a vesicle,
yeah,
which could
Muscle.
stay in the cytoplasm.
or it's gonna
It
go
can
straight to the Golgi body. So what the Golgi body does,
it
it's like-
transforms the protein chain a little bit more.
and then it's gonna turn the protein into its final form, and then it's gonna go.
And it'll come to the end of the Golgi body as well. And then-
Exocytosis.
exocytosis. And then it can still stay within the cell, or it could go to
You can
blood-
leave the cell.
it could leave the cell and go to the blood, which then it becomes a hormone.
Yeah.
And then this is rough ER, which is where it reads it, and then the smooth ER is where it forms membrane.
So rough ER is basically involved in protein synthesis. The smooth ER, this other stuff. This is a free ribosome. It's just another ribosome that the mRNA can go through.
If it goes through a free ribosome, it stays within the cell. So the protein chain will just stay within the cell.
What about rRNA? rRNA is the RNA inside the ribosome.
Yeah.
I believe.
Okay, next.
Did we mention everything?
Yeah, I think so.
Okay.
So,
Cell division.
So we have these things called
Centrioles.
centrioles. All right. So centrioles, during cell division, everything, the cell prepares itself for cell division by doubling up on
Duplicating
every.
everything.
So let's just pretend we have two DNA things here.
Yeah.
Just pretend we have a chromosome. In these. Okay.
So the centrioles, they duplicate. So one goes to one
That's
one
too
side
far.
of the cell.
I can't see it. It's off camera.
It's
There it is.
it's over there, and then the other one goes to the other side of the cell, and then the microtubules attach to centromeres, and
Of
then
the DNA.
of the DNA,
Mhm.
and then the centrioles they pull the DNA, the centromeres to opposite sides of the cell.
They split up the chromosome.
Yeah,
So that.
and then all the organelles are divided into the two new daughter cells through mitosis.
No, stop.
And the endomembrane system means that all the membranes of the organelles within the cells are all connected because they all
They all come from the same.
yeah
thing.
they all come from the same thing.
They are pretty much the same thing. They're all just phospholipid bilayers.
Yes.
Yeah.
And the vesicles don't move to other places like because of a floating freeway. They move due to the cytoskeleton or
Yeah, they move
the
along.
microtubules.
They will
the
move with the
microtubules.
guidance.
Yeah.
These are microtubules right here.
They're all connected.
Yes, everything is connected. Okay, are we done?
We're done.
We're done.